Welcome to the Lecture 4 Basics video. Let's learn a bit more about the plasma membrane of the cell. You already know about the lipid bilayer formed by phospholipid molecules, but there's more going on than just phospholipids. There are many different types of lipids, proteins, and carbohydrates associated with the plasma membrane. So let's take a look at each. Lipids are defined as large molecules that mix poorly with water. Here are several examples of membrane lipids. The phospholipid you've seen before, but membranes also have ceramides, cholesterol, and sphingolipids, just to name a few. And you should see the similarity between all these molecules. They're all amphipathic and have polar heads and long nonpolar tails. The proportions of different lipids is different in different cells. In order to understand different proteins, we need a quick review of protein structure. All proteins are essentially a ribbon of amino acids. Every amino acid has an amino group and a carboxyl group, and then an R group that makes it different from other amino acids. That's what makes a chain of amino acids be a chain of glycine, proline, threonine, and so on. This ribbon then folds up into a complex shape: alpha helixes, beta pleated sheets, secondary, tertiary structure, quaternary structure, and this forms a complex shape depending on what R groups are present and whether they're polar or nonpolar. All proteins are made of these same building blocks, but end up looking very different. Here's a model of an aquaporin channel, for instance. You can see each of the amino acids in this diagram and see that the protein folds up to be mostly inside the membrane. You would expect nonpolar amino acids to be found inside the bilayer and polar amino acids to either be exposed to the cytosol inside the cell or exposed to the extracellular fluid outside the cell. Membrane proteins have many different shapes and functions. Some are transporters or act as enzymes or are receptors like this one, or recognize other cells or hold cells together or attach the membrane to the extracellular matrix. And you'll see several examples of those in this lecture and the ones near it. Carbohydrates, or sugars, are compounds with very fixed ratios of carbon, hydrogen, and oxygen. They generally form a ring structure and can either be monosaccharides or disaccharides or polysaccharides. The term glyco, it means that a carbohydrate is involved in the molecule. Since carbohydrates are polar, they're not found within the lipid bilayer. Instead, they are bound to either lipids or proteins on the outside of the cell. Here's an image to demonstrate that. Carbohydrates are shown bound to lipids, which create glycolipids, and shown bound to proteins, creating glycoproteins. These are believed to form some sort of cell coat or barrier that protects the cell and may also act as adhesion points to allow other cells to bind to each other. In addition to the membrane itself is the extracellular matrix. This group of carbohydrates and proteins holds cells together and allows them to migrate during development. I'm going to have you use your book to label this diagram and write down these simplified definitions of the 4 important molecules of the extracellular matrix. So your notes should contain information about all these structures. Be sure that you've got them written out and use your book glossary to define any differences between the terms so we can discuss them in class. Another good study tool is to redraw an image like this one just in cartoon form and be able to label it from memory. Lastly, let's look at the fluidity of a plasma membrane. Proteins need to be able to move within the membrane, and here's a couple of examples. We've already seen the sodium-potassium pump, and it has to change shape in order to allow sodiums to be pumped out of the cell and potassiums to be brought into the cell. That means the protein has to be able to move within the lipid bilayer. Here's another example that's called a receptor tyrosine kinase, and we'll see that more later in the cell communication lecture. But notice that it has 2 membrane proteins, and when they bind to an external stimulus, the 2 proteins have to move together within the membrane and dimerize or bind together, and this requires that they be able to move within the lipids and in a way that makes the lipids fluid. So there's three factors that we're going to talk about in this class that determine membrane fluidity, or how easily things move around within the membrane. And the first is the level of saturated phospholipids and unsaturated phospholipids. Saturation refers to the chemical structure of the phospholipid tail, specifically how many hydrogens are bound to the carbons in the tail. If each carbon has two hydrogens, the tail is called saturated with hydrogens, as you can see here, and is a very straight tail. If a carbon, however, has a double bond, instead of some hydrogens, it becomes unsaturated, it's not full of hydrogens, and this makes a kink in the phospholipid tail. So as you can see, a membrane with a lot of unsaturated phospholipids is going to be more loosely packed and therefore more fluid. Second, temperature affects how fluid a membrane is. You learned in high school physics that increasing the temperature of a system increases the kinetic energy of the molecules, and they randomly move faster. This increases fluidity. Lastly, cholesterol is an amphipathic lipid with a small polar head and a stiff ring tail. Its shape means that the phospholipids cannot move as freely in the membrane when there's a lot of cholesterol around. So at normal temperatures, more cholesterol inhibits fluidity. But at very cold temperatures, where membranes normally crystallize or gel, cholesterol prevents the freezing of the membrane by physically blocking the tight packing that phospholipids would otherwise do. This is important to exothermic organisms more than to everyday mammals. That's it for this video. You should be able to do the following before taking the basics quiz. Thanks for watching.